This is the Space Habitat event. Now we'll have the panel about food production, and then we'll have the presentation of Federico Granados Unger about the using biotechnology for food production on Mars.
Okay, thank you very much. So thank you, first of all, for this invitation to participate in the Space Habitat event. I'm honored to be part of it. So in this presentation, I'll talk about what's the role of biotechnology for food production on Mars, and why is it so important for the future of human colonization in the Red Planet. Okay, so for this presentation, I'll give some insights of a previous presentation that I did as part of a member of the Mission 50 at Habitat Marte, one of the many virtual missions that have been taking part in this event. So I hope you enjoy it. Okay. It's important to know that for this for this mission, we were all assigned different facilities. So each one of us during the mission were the chiefs of that facility, and my facility in that case was the greenhouse. So all of these perspectives is I will be talking about this facility, about how would be a greenhouse in Mars. Okay, so first of all, talking about the internal design of the facility. First, I'll talk about the whole station that's composed of several different facilities. It was proposed in a previous virtual mission by Josh Rathod. It included, this includes nuclear power, the power station, a solar farm, MOXIE for the production of oxygen, an engineering facility, sanitation center, a main station for regulating the activities of all the other facilities, a launch center, a health facility, and the greenhouse, which is here, and it's connected in an important way with other facilities like the engineering facility, sanitation center, the main station, and the health facility. It collaborates mainly with these facilities. Here's another point of view of where would the greenhouse facility would be. And well, for this facility, we took into account 8 meters of diameter and 3 floors, where we would fit all of the different requirements of the station, right? For example, in the bottom floor, a lab and a workspace would be. Then on the second floor, there would be a cultivation area, and on the third floor, an area for social and entertainment, sleeping and toilet, and a kitchen, right? Among other things. So this is like a detailed internal design of this facility. Well, from left to right, there's the first floor, then the second, and the last one is the third floor. And here we included, for example, stairs, a sanitation hall for the incoming astronauts, the ECLSS for the purification of water and oxygen, an MMRTG for the production of heat and electricity, a maintenance area, a control area, a dry lab, and 2 subdivisions of wet labs that I'll talk about next, storage area, processing area, a urine processing device, traditional cultivation area, an ethylene scrubber that I'll talk as well next, robot storage area, an aeroponic culture area, bioreactor and photobioreactor area for bacteria and algae, WC, dorms, kitchen, dining room with social area, and a vertical garden. So this facility would be able to fit in the cargo area of the SpaceX Starship rocket, so it can be taken to Mars in the future. So what are some of the challenges for the growth of plants in Mars? On Mars, first of all, we have a great difficulty in capturing like all of the chemical characteristics of the Martian surface in a single Mars soil simulant, which could be used for the growth of plants in the red planet. In addition to this, it has been stated by this paper that, for example, to know how will the crops perform in the red planet, we will require a lot more basic knowledge or fundamental knowledge on plant biology. So because we still don't know many things about, for example, how is the diffusion of CO2 from the plant, or how does the plant breathe from the roots. And this is very important to know if it's viable to grow plants on Mars directly on the regolith with the current technology. So we had to develop new technologies for the growth of plants. In addition to this, it's very cold. The average temperature on Mars is of minus 55 degrees Celsius, because it's, I think, the planet that gets the least sunlight from the rocky planets. It has a very thin atmosphere, which offers no protection against cosmic radiation, or very few, which could be very harmful for all of the life forms, or at least the complex life forms that will live on the planet. In addition to this, radiation can turn certain compounds in the Mars surface to toxic perchlorate cells, which are already present in the whole surface of the planet, which is very dangerous for future settlers, because if plants that are grown directly in the soil take up these salts, and after that the settlers consume them, these salts can cause them hypothyroidism, which is very dangerous for their health, in addition to other diseases. In addition to this, the soil the Mars regolith has very low nitrogen content, which is bad for the growth of plants. And CO2, yeah, it's widely present in the planet, but at a very low concentration. So plants have a very hard time taking them for their photosynthesis. In addition to this, well, we also have heavy metals that are poisonous, and they can be widely available in the soil of Mars. And there's a high difficulty, for example, in cultivating high culture tubercules, such as potatoes, either in hydroculture or in analog soils. This is very worrying for future settlers, because these potatoes, for example, would be very helpful for these settlers in order to have the sufficient energy to perform their tasks. So we should find ways to grow them in the planet. So some of the technologies I proposed in order to solve some of these challenges is, for example, to include 2 labs, a dry lab and a wet lab. For the subdivision of the wet lab would be a molecular biology lab, which could help scientists to obtain, for example, DNA and proteins and metabolites from plants, which could be, for example, in the case of genes, these genes could be sequenced and then passed on to the dry lab, to the bioinformatics lab, which consists only of computers, for analysis of which genes are being expressed in these plants, which could point out which problems are they having, right, when growing in the planet. So we by using these 2 labs, we could know what changes have happened to these plants and why does they occur. For example, lignin content. We have an analogy for humans. Humans, their support are bones, right? It has been shown that in microgravity, like in the International Space Station, their bones, the composition of the bones change a lot in microgravity. They become more porous, and astronauts have problems with their bones in microgravity. So plants may have problems as well in their own support, that is lignin, which helps them stand upright. With their culture in microgravity, right? We know that Mars has 1/3 of the gravity of Earth, so it may be a problem for them. In addition to this, low gravity has also been shown to have oxidative stress to plants, which can damage the DNA repairing machinery, which could possibly be fatal for them, right? Also, microgravity can affect their immune system, as it has been shown that some genes related to this system turn on and others turn off, which can hinder their ability to fight off pathogens. And also, there are a lot of photosynthetic inefficiencies of plants. And for example, they are very slow in converting carbon dioxide to sugars for them, or even to adapt to light fluctuations, right? So with this basic knowledge of plants that would be acquired using these two labs, selected species that would be useful for the settlers can then be taken, for example, to the tissue culture lab for their enhancement, so they can better adapt to the conditions in the red planet. This is the other part of the wet lab that I proposed, a tissue culture lab, which could be used for to genetic engineer these plants in order to enhance them, to make them more resistant to the adverse conditions of Mars. And it can also be used to obtain several drugs of interest from them. In addition to this, another biotechnological technique called in vitro micropropagation can be used in order to obtain clones of plants. For example, the Paulownia tree, which I talk next, which could be very useful for terraformation efforts. It has already been micropropagated here in Mexico in worldwide first. So this is a very exciting technique. Some advantages of it is that it needs a very short time to obtain new plantlets. It can generate a lot of plants that are free from viruses, so that you can obtain relatively safe plants. It allows the culture of a great number of them in a very small area. Like, for example, you can obtain 1,000 plants in just one square meter, and it proves a lot more successful in cultivating challenging plants when you compare them to traditional soil. So this is a very interesting approach. And in addition to this, as I just mentioned, potatoes, for example, have some problems growing in hydroponics. So another option is using aeroponics to use this kind of plants, where using this technique, plants are grown in a misty air environment that provides them with water and nutrients, which you can see, for example, in the bottom image. And for example, in order to fight the lack of nitrogen in the soil, bioreactors in this facility can be used to culture nitrogen-fixing bacteria, which could be able to fix atmospheric nitrogen from the Martian atmosphere, and also to eliminate the toxic perchlorate salts in the Mars soil, so it can be made safe for human use, and it can be more useful for plants to grow. There are other types of equipment that are proposed, but we are short of time, so I won't talk about them. And some of the plants that are proposed, like, for example, to be cultured in this system in order to provide the nutrients for humans are these that according to this paper, if you grow soybeans, spinach, mushrooms, wheat, seaweed, and you complement them with this strain of algae, Spirulina platensis, which can be cultured in photobioreactors. By culturing these plants and algae, you can provide all the nutrients that humans need, both adults and children, for a healthy 2,000 kilocalorie diet. So these are the plans that I proposed. And for finalizing this talk, for example, for terraforming efforts, we can use, as I mentioned, in vitro micropropagation of the Paulownia tree that can also be further enhanced using the labs proposed and the techniques proposed. And this is very interesting because it's the tree that grows the fastest in the whole world. It has a very light but strong wood. It's considered semi-precious wood. It helps regenerate contaminated and eroded soils, and is the tree that absorbs the most CO2 in the world, 10 times better than any other tree, which can really help us in terraforming efforts. We could also perform the cultivation of bluegrass of the genus Poa, which can handle frigid temperatures and intense solar radiations, which is the characteristics that we would need for plants to be grown on Mars because of its harsh atmospheric conditions. So they're perfect for that, and they take little space, require little maintenance, and produce lots of seeds. We could also, for example, in order to make certain regions of the planet surface more better for growth of plants for terraforming efforts, we can use sheets of silica aerogel, which can be placed in those regions, and this can warm the ground, melt the ice in order to obtain liquid water and help it make an environment suitable for plants. So these are some strategies that we could use that are aided by biotechnology in order to support the terraforming efforts. And well, this is all from myself. And thank you very much for this time.